I can do to help you? Yes! Thank goodness you're here, Nitrate Reductase. My backpack is just too heavy with these great oxygens on it.
It's okay. I can help you. Let me see your backpack.
Man, it can be nitrite. Yay! Thank you, Nitrate Reductase. Yay!
Okay, now just to break that down a little simpler for everybody. Nitrate reductase is an enzyme that catalyzes interactions in the cytosol of plant cells. Its function is to reduce nitrate to nitrite. Nitrate reductase does this by removing an oxygen from the nitrate. This process makes nitrate reductase very important to the life cycle of plants. But hold on to this thought because we'll talk about this a little later on in the video.
Nitrogen reductase is a flavoprotein. Flavoproteins are proteins that contain either flavin adenine dinucleotide or flavin mononucleotide, both of which are derivatives of riboflavin. Nitrogen reductase contains flavin adenine dinucleotide, or FAD, a heme group, and a molybdenum. In the picture being shown, the molybdenum is highlighted in yellow inside the nitrate reductase structure, which is pink. The molybdenum is responsible for breaking the bonds between molecular nitrogen to allow for nitrogen fixation in plants.
Heme is a prosthetic group that binds iron. Here in the image you can see both of the heme groups in yellow on the left hand side of the image. The iron in the heme group allows for an oxygen to bind. But once the oxygen binds, the nitrate is reduced to nitrite as said earlier. Here in this picture, we can see the iron in the center of the 4-pyro ring linked by methane bridges. The reaction being shown shows nitrate reductase working in the opposite direction, taking nitrite to nitrate.
The image you see now is the nitrate reductase enzyme in the bacteria E. coli. Nitrate reductase in E. coli is a homodimer that has 1,246 amino acids. Cysteine 222 is an essential amino acid because it is responsible for the binding of the molybdenum to make the redox center of the enzyme functional. Histidine 56 and 66 are also essential in nitrate reductase because they bind the heme groups.
Look guys, it's really simple. In the cytosol of plant cells, what happens is nitrate reductase reduces nitrate to nitrite. This is the preliminary step of the electron transport chain that makes up the denitrification process.
Nitrate reductase is thought to have 2 active states and 1 inactive state. In the 2 active states, there are 2 active sites. The first active site binds NADH and NAD+. The second active site binds nitrate and nitrite.
At the first active site, NADH reduces FAD and passes the electrons to the molybdenum. At the second active site, the nitrate is reduced to nitrite.
Nitrate reductase is controlled by the plant's metabolic activity. The enzyme is induced into activity based upon the availability of nitrate and the amount of light in the cytoplasm.
Glutamine levels also influence the activity of nitrate reductase.
Glutamine levels are low. Nitrogen reductase and the nitrogen reducing capacity are increased.
If the glutamine levels are high, the activity of nitrate reductase is decreased. Look over there. It's an innocent nitrate in trouble.
Is there anything I can do to help you? Yes! Thank goodness you're here, Nitrate Reductase. My backpack is just too heavy with these great oxygens on it.
It's okay. I can help you. Let me see your backpack.
Man, it can be nitrite. Yay! Thank you, Nitrate Reductase. Yay!